Hello everyone. So today we'll be talking about Agrobacterium. So Agrobacterium is nothing but the nature's genetic engineer. It has the ability to transfer a part of its DNA into the genome of the plant and use the plant to provide nutrients for its survival. Based on this remarkable Agrobacterium system, scientists have developed a powerful tool for transformation. So before we begin, let's look at a few keywords. First being plasmid. So a plasmid is a small extra chromosomal DNA molecule in the bacteria that replicates independently of the chromosomal DNA. So a few wild agrobacterium components would be firstly the tDNA region. It carries oncogenes like auxin, cytokinin and opine genes. They induce tumor-like growth in plants. Second would be opines. They are amino acid derivatives that serve as a carbon source for agrobacterium. Third would be the virulence genes. They are essential for tDNA transfer into the plant cells. So here we can see a diagram of the TI plasmid, and it contains the tDNA region on the upper side which gets transformed into the plant cells, and we see its auxin, cytokinin and opine producing genes. On the lower side we see the virulence region, and most importantly the ORI, which is origin of replication. Our next keyword is a vector. So a vector is a vehicle or a medium. It is a carrier that transfers DNA from one cell to another. It helps in genetic engineering by inserting new genes into organisms. Examples include plasmids, viral vectors, and cloning vectors. Vectors are used to create transgenic plants and animals or to study genes and proteins. Here we see a diagram of the Agrobacterium TI plasmid being used as a vector, which we will elaborate further. Our next keyword is Agrobacterium as a vector. So, scientists have modified Agrobacterium's TI plasmid by removing tumor causing genes, thus creating a safe version for the genetic engineering. This disarmed agrobacterium is now widely used as a vector to transfer desired genes into plants. Many crops and horticultural species are routinely transformed using this method, and its applications continue to expand. Here we can see the mechanism, and the mechanism starts with detection of wounded plant signals. Secondly, the agrobacterium would activate its virulence genes in its TI plasmid. Then it cuts out the tDNA and prepares it for the transfer. Next up, it injects the tDNA into the plant cell through a secretion system, which will elaborate further. Next up, the tDNA integrates into the plant's genome, permanently changing it. Next, the plant produces tumors and opines, benefiting the bacterium but not the plant. And lastly, we know how scientists have been modifying this system to insert useful genes instead of the harmful ones. So the whole process can be summarized using 4 steps. First being recognizing a host. So the agrobacterium targets wounded plant cells by detecting phenolic compounds like acetosyringone, which are released upon injury. The VIRA and VIRG proteins sense these signals, guiding the bacterium to the wound via chemotaxis. It then attaches firmly to the plant cells using surface proteins and polysaccharides. The second step would be activating the TI plasmid. So, Agrobacterium uses its TI plasmid to transfer tDNA into the plant genome. The tDNA induces tumor growth and opine production, benefiting the bacterium, as discussed before. The VIR region aids this transfer, activating in response to the plant wound signals. The VIRA detects acetosyringone, triggering VIRG phosphorylation, which in turn activates other VIR genes. The VIR-D2 excises tDNA while VIR-E2 protects it during transfer. The third step would be transferring the DNA. So the agrobacterium injects tDNA into the plant cell via a type 4 secretion system. The VIR-D2 and VIR-E2 guide the tDNA into the cytoplasm, where the plant's nuclear machinery transports it to the nucleus. There, the tDNA integrates into the genome, making the plant follow its new genetic instructions. And last step would be plant manipulation. The inserted tDNA disrupts plant functions, leading to first off tumor formation. Overproduction of auxins and cytokinins causes uncontrolled cell division, forming crown gall tumors. Second would be opine production. The plant produces opines, unique nutrients that only agrobacterium can metabolize, giving it a competitive advantage. So now let's look at the key features of agrobacterium mediated gene transfer. So first would be its natural gene transfer capability. Agrobacterium is one of the few bacteria that can naturally transfer DNA into a plant genome. It enters through plant wounds, detects signaling molecules like acetylsyringone as discussed before, and activates virulence genes. The bacterium then transfers tDNA into the plant nucleus, causing genetic transformation. As an example, scientists use this process to introduce beneficial traits like pest resistance and drought tolerance in crops like soybean, tomato, and rice. Secondly, the TI plasmid as an engine of genetic transfer. So the TI plasmid is essential for agrobacterium's ability to transfer gene. This includes first off tDNA, the DNA segment inserted into the plant carrying new traits. Secondly, the VIR genes, helping process and transferring tDNA into the plant. Thirdly, the opine catabolism genes, allowing agrobacterium to use opines as food. And last but not the least, origin of replication, which enables the plasmid to replicate inside the bacterium. As an example, in golden rice, a modified TI plasmid was used to insert genes for beta carotene production, enhancing its nutritional value. Third would be host specificity and range. So, agrobacterium naturally infects dicot plants like tobacco, potatoes, and tomatoes, but it was less effective in monocots like wheat, rice, and maize. So, advances in biotech have expanded its use in monocots. So, why were monocots resistant? It's because they have weaker wounding response, making bacterial attachment harder. They have lower production of phenolic compounds like acetylsyringone, which activate the agrobacterium. Thirdly, they have thicker and more rigid cell walls, making the DNA transfer difficult. The solutions for this would be pre-treating monocots with acetylsyringone to trigger infection. Also, modifying tDNA and VIR genes to enhance DNA transfer. As an example, scientists have used agrobacterium to insert herbicide resistance genes into wheat and rice, overcoming the monocot barrier. Fourth would be stable integration into the host genome. So, unlike temporary gene expression, agrobacterium ensures permanent integration of genes into the plant's DNA. So, why is stability important? It is because the gene is passed down to future plant generations. It ensures long-term consistent trait expression. It reduces the need for repeated modifications, saving time and cost. For example, BT cotton, modified for pest resistance, remains effective across multiple growing seasons due to stable gene integration using agrobacterium. Fifth one would be opine catabolism. So, agrobacterium tricks plant cells into producing opines, which are nutrients that only the bacterium can utilize. This is significant because it ensures agrobacterium survival by making the plant produce excessive nutrients. It gives Agrobacterium a competitive edge over other soil microbes as discussed before. So scientists have developed opine-free strains for genetic engineering to avoid this side effect. As an example, crown gall tumors in infected plants produce nopaline and octopine, which Agrobacterium metabolizes, securing its dominance. Sixth would be the type 4 secretion system. So, Agrobacterium employs a type 4 secretion system, a specialized molecular syringe to transfer DNA into plant cells. The key steps in tDNA transfer are the VIRD2 cutting the tDNA from the TI plasmid, and VIRD2 and VIRE2 coating and protecting the tDNA as it moves through the bacterial membrane. And lastly, tDNA entering the plant cell and integrating into the genome. As an example, the T4SS is also found in human pathogens like Helicobacter pylori, highlighting its evolution and evolutionary significance in DNA transfer. This can be further understood using the links given below in the description. Here is a table displaying the minimal cellular damage as compared to other methods like electroporation and gene gun, where Agrobacterium shows lower cell damage, lower DNA fragmentation, higher integration stability, and higher efficiency as compared to other methods. And lastly, we see long-term gene expression. So, Agrobacterium enables long-term gene expression by integrating DNA into a plant's genome, allowing traits to be inherited. So, the ongoing research is about CRISPR Agrobacterium fusion for precise gene editing and expanding its effectiveness in monocots. Also, developing safer strains to prevent unintended gene transfer. For example, the future applications include engineering drought-resistant crops to address the climate change. Here we see a table again displaying the characteristics of Agrobacterium as compared to other methods like electroporation, gene gun, and viral vectors, showing how Agrobacterium mediated gene transfer is still one of the best methods for biotech. Next, we shall talk about cell competency. So, cell competency refers to a cell's ability to take up foreign DNA from its environment. This is a crucial step in genetic transformation, allowing scientists to introduce new genes into the cells. So, firstly, the cell competency depends on transformation efficiency, using highly competent agrobacterium cells to maximize tDNA uptake and successful gene transfer. Secondly, antibiotic resistance, ensuring the antibiotic resistance of your agrobacterium strain is different from the resistance marker on your vector to avoid selection issues. Third would be compatibility with the plants. So, choosing an agrobacterium strain proven to work with your target plant. Following established protocols as plant susceptibility, gene delivery efficiency, and regeneration ability, all of these factors impact the success of gene transfer. So, here we have elaborated on the second point, which is antibiotic resistance as a factor for cell incompatibility or compatibility. The EHA 101 strain is kanamycin resistant, but the PBIN19 vector also uses kanamycin as a selection marker. So, this makes it difficult to distinguish between transformed and non-transformed cells. So, the solution is using the EHA 105 strain, which is rifampicin resistant instead. Applying kanamycin plus rifampicin together to select the only cells that were successfully taking up the vector was the key idea here. So, choosing an agrobacterium strain with the same antibiotic resistance as the T binary vector leads to the growth of both transformed and non-transformed cells, reducing transformation efficiency. So here we see an image displaying agrobacterium mediated transformation. So the step 1 would be extraction of the TI plasmid from Agrobacterium tumefaciens. So what happens is Agrobacterium tumefaciens, a naturally occurring bacterium, harbors a TI plasmid, which is the tumor inducing plasmid. This is all we've learned before. In nature, this bacteria would infect wounded plants, transferring its tDNA into the plant cells, causing tumors, that is, crown gall disease. So, what scientists do is they extract the TI plasmid from Agrobacterium. Instead of using its natural tumor causing genes, they modify the tDNA region to carry a gene of interest, for example, resistance to pests and improving crop yield. The second step would be cutting the TI plasmid using a restriction enzyme. So what happens is the TI plasmid is treated with restriction enzymes. These are molecular scissors that cut DNA at specific sites, and a precise cut is made in the tDNA region where the new gene will be inserted. This is necessary because this allows for the replacement of tumor inducing genes with a desired gene, ensuring that the plant does not develop tumors but instead gains beneficial traits. The third step would be inserting the foreign gene into the TI plasmid. So what happens is a foreign gene, for example drought resistance and pest resistance, is cut from a donor DNA source using the same restriction enzyme. This gene is then ligated or joined into the cut TI plasmid using DNA ligase, which is nothing else but the DNA glue enzyme. The result is a recombinant TI plasmid carrying the desired gene instead of the original tumor inducing genes. Here, the key consideration is that the foreign gene is inserted between the specific border sequences, left and right DNA borders, ensuring that only the desired gene is transferred into the plant. Also, a marker gene, example antibiotic resistance, is also inserted to help identify successfully transformed cells later. The fourth step is reintroducing the recombinant TI plasmid into Agrobacterium. So what happens is the modified TI plasmid, now carrying the gene, the desired gene, is reintroduced into Agrobacterium using electroporation or heat shock methods. The bacterium now acts as a gene delivery system capable of transferring this foreign DNA into plant cells. So we see why only Agrobacterium is used. It's because it has a natural ability to transfer DNA into plant cells through a process called horizontal gene transfer. It enters plant cells via a type 4 secretion system, which allows the direct injection of tDNA into the plant's nucleus. The step 5 would be plant cell transformation or co-cultivation of Agrobacterium with plant tissue. So how is this transformation done? The transformed Agrobacterium is mixed with plant tissues, example leaf discs, stem segments or callus cultures. This mixture is placed in a co-cultivation medium where the bacteria attach to plant cells and transfer the tDNA. The tDNA integrates into the plant's chromosomal DNA, making the plant genetically modified. So what happens inside the plant cell is that the tDNA is transferred into the plant nucleus via a molecular transport mechanism. So the foreign gene integrates into the plant's genome where it will be stably inherited by future plant generations. The step 6 or the last step would be selection and regeneration of transgenic plants. So after the transformation, the plant cells are transferred to a selection medium containing antibiotics or herbicides. Only the successfully transformed cells, those that receive the foreign gene, will survive thanks to the marker gene, that is antibiotic resistance. So for plant generation, the plant regeneration, the surviving plant cells are induced to differentiate and grow into full plants using plant hormones. This can occur through organogenesis, that is root and shoot formation, or somatic embryogenesis, which is artificial seed formation. So here the key takeaway is scientists modifying its TI plasmid, replacing tumor-inducing genes with beneficial ones. Secondly, modifying modified tDNA, which integrates into the plant genome, making the plant transgenic. Thirdly, a selection process which ensures that only the successfully transformed cells have survived. And lastly, the final product, which is a genetically modified plant with desirable traits. And like any other aspect of healthcare, agrobacterium mediated gene transfer also carries its own nuances and limitations. So the first one is host range limitation. So here the challenge is that it works well with dicots, example tomatoes, soybeans and cotton, but it's less efficient in monocots like wheat, maize and rice. This is due to lack of natural signaling molecules for agrobacterium infection and thicker cell walls, making DNA transfer difficult. So the solution would be using supervirulent strains like EHA 105, LBA 4404, which are designed for monocots. The wound plant tissues, which co-cultivate with helper bacteria to boost infection, can also be used. Thirdly, we can modify transformation conditions with monocot adapted promoters and agrobacterium accessory proteins. The impact is that these strategies improve transformation efficiency in monocots but may still be less effective than direct methods like ballistics or gene gun technology. The second limitation is gene size restriction. So here the challenge is that the tDNA region in the TI plasmid has limited capacity for foreign DNA. Large genes or multiple gene insertions are difficult. So the solution would be using binary vector systems with disarmed TI plasmids for larger insert capacity. Secondly, using multiple tDNA insertion strategies to introduce multiple genes in separate events. Thirdly, using engineered vectors designed to carry larger DNA fragments efficiently. Here the impact is that these approaches help expand the range of genetic modifications, though very large inserts may still be requiring alternative methods like transposon based systems or artificial chromosomes. The third nuance is low transformation efficiency for some species. So here the challenge is that some plant species, especially certain monocots and specific genotypes, are highly recalcitrant to agrobacterium mediated transformation. Efficiency depends on factors like plant tissue type, bacterial strains and transformation conditions. The solution would be to use plant specific promoters to enhance gene expression using stress treatments like osmotic pressure, chemical signaling like acetylsyrinzone to improve transformation efficiency. And thirdly, using co-cultivation with helper bacteria to assist agrobacterium infection. Here the impact is that by optimizing protocols for each species, transformation efficiency can be significantly improved. This enables successful genetic modification of previously resistant plants. The fourth nuance is how time consuming and labor intensive it is. So the challenge is that the transformation process from DNA transfer to plant regeneration takes from weeks to months. It requires sterile tissue culture conditions and skilled handling. Gene gun and biolistic methods are faster but lead to lower stability of gene integration. So the solutions would be using direct tissue transformation techniques to bypass lengthy tissue culture steps. Also using optimized plant regeneration media to speed up growth and recovery. Thirdly, using in planta transformation, that is direct gene transfer into seeds or seedlings to avoid prolonged callus formation. Here, the impact is that faster transformation methods can accelerate research and commercial applications, making genetic engineering more efficient. The fifth nuance is the risk of random tDNA insertion. So, the challenge is that tDNA may integrate randomly into the plant genome. It may disrupt essential genes or cause unexpected mutations, which would result in variable gene expression, making outcomes unpredictable. So, the solution would be combining CRISPR-Cas9 with Agrobacterium for precise genome editing and modifying tDNA border sequences to target specific chromosomal sites. Here, the impact is that more controlled insertion can be used to reduce unwanted mutations and ensuring stable gene expression. The sixth nuance is silencing an unstable gene expression. So, the challenge is that plants' epigenetic mechanisms can silence the integrated gene. Methylation of the promoter region may block gene expression. Position effects can lead to inconsistent or weak expression. So, the solution would be using strong constitutive promoters like CAMV35S, ubiquitin, and actin 1 to maintain the expression. Also, applying RNA interference, RNAi suppression strategies to counteract this gene silencing, and incorporating insulator sequences to protect the gene from epigenetic suppression. Here, the impact is prevention of loss of gene function over time, ensuring stable and reliable trait expression in transgenic plants. The seventh nuance is regulatory and ethical concerns, which is challenging because there are strict regulations for genetically modified crops in many countries. There are public concerns over safety, environmental risks, and unintended effects. Also, it is a lengthy and expensive process for getting approval and commercialization. So, the solution to this would be cisgenic approaches using genes from the same species to reduce regulatory hurdles, using non GMO alternatives like transient gene expression to bypass strict regulations. Also, providing public education campaigns to improve awareness and acceptance of GM crops. Its impact is that it eases commercialization, improves public trust, and accelerates the adoption of biotech crops. And last but not the least nuance is limited success in woody plants and gymnosperms. So the challenge is that Agrobacterium struggles with woody plants and gymnosperms due to thicker cell walls and lower regeneration capacity. So standard transformation methods have low efficiency in these plant types. So the solution would be using somatic embryogenesis to improve plant tissue culture regeneration, using cell wall degrading enzyme modifications to enzyme bacterial entry, and using root based transformation for better gene delivery. The impact is that it enhances transformation success in previously resistant species, expanding genetic engineering possibilities. So, this was all about Agrobacterium, and thank you for watching.